right. Hey, Nigam.
Hi, Julia.
Hey. Yeah, so I guess Nandu said he just didn't maybe see that email come in initially, so he didn't have the link. But
It's definitely there, but I'm gonna send it to him again.
perfect. Perfect. Yeah, I'm sure it just got buried in his inbox.
Cool. We had a lot of people register.
Yeah? Oh, that's exciting. How many?
58.
Oh, nice. That is a good number.
It's pretty high.
Yeah, that is. That's higher than average, right? For
Yeah,
this.
it's like double.
Nice. That's good to hear. How are you doing?
I'm okay. What about you?
Pretty good.
In a lab for you these days, huh?
Not much, no. So we've been asked to work from home as much as possible. So it's pretty nice, actually. I'm not complaining. Although if I had to do this for the past 9 months, I think I'd be pretty sick of it. But
Yeah, I mean, don't yeah,
mm-hmm.
give it a little bit.
Yeah, exactly.
So like crucial things when Nandu gets on are to just make sure that the PowerPoint switch thing will go smoothly.
Right. Yeah, that was what I was gonna ask you about. And then also, I guess just making sure I understand how to bring up my PowerPoint, I guess cohesively, or maybe I should just do that right now, right? And then we can walk through what it'll be like when we switch.
Yeah, I can like kind of help coordinate a little bit, and I think
Okay,
Nandu's with us.
perfect. Hi, Nandu.
Hi, hi, Nigam. Hi, Julia.
Yeah, awesome. Okay.
Hey,
So,
it's great. Nandu, I can't see you, but maybe it's just because I'm on the
oh, is it?
browser. But it's great to meet you, though. I know we've emailed a lot. I'm happy to finally connect with you.
Oh, yes, yes, same from here. But my video is on, so I don't see myself on the screen, so
Yeah, I
I
can't see
don't
you
know.
either.
That is interesting.
Maybe are your video settings within Zoom set to the right camera?
I don't change the setting. I use Zoom quite a bit.
Okay. Yeah,
I wonder
it's odd.
why. Interesting. Yeah, there's only one.
Yeah, because I see it says your, let's see, spotlight video, make host, change filter.
Let me log off and log on again.
Yeah, I
Then
think that's the probably smoothest thing to try. Yeah, we'll be here.
Okay, perfect. And in the meantime,
Is your internet fast now, Julia?
yeah, it's really fast now. So,
Cool.
yeah.
Part of the reason, like, we used to try to do this when we had like enough bodies was to like have this kind of technical backup, which I'm kind of like serving as today. So
Yeah, right. Okay. Oh, there you are,
okay,
Nandu.
now
You can
you
see you
can
now.
see me. I was logged on to the Google platform, so that
oh.
explains.
Well, hey, it's nice to see you now, Nandu. I can see your face. It's great to see you.
Right. That's
I know.
right. I think hopefully once this COVID is done, we should be able to meet sometime.
Right.
Yeah,
Yes,
let's get together. That would be great.
Yeah.
absolutely. Okay, so right now, are you seeing my presenter view or the actual PowerPoint display view?
Yeah,
I
so we're seeing your presenter view, which you
Okay.
don't really want to show. You
No.
want to, yeah.
Yeah. Okay, perfect.
so,
And
so,
then,
so
oh yeah,
Julia, could I share my screen? That way, if I need to go to the original paper for any description or show the USP landing page for cannabis for any links, that would be helpful.
yeah, absolutely. So Nigam, I'll let Nigam jump in here because he's the expert at doing this, but I believe I'll start with my intro slides and then I will switch the screen share over to you. And Nigam, do you have any?
Yeah, so let's just walk through it. So it'll go. So I know you guys have like talked about the flow or whatever, but presumably it'll start here. Julia will, will open the webinar for attendees to come in right at 11 and 11 Pacific in 7 minutes. And then maybe one after, Julia welcomes everyone. She'll introduce you, she can read your bio if that's smooth. And then Julia, can you go ahead and tab through the next couple slides?
Mm-hmm.
So then Julia will give some CANN announcements. Then she's going to thank our sponsors. And then we will, Julie, can you go ahead and tab through the next one or two? Right, yep. So thanks to sponsors, and then here, and then this is where we'll switch. So then Nandu, do you have an option to share your screen?
Yes.
Okay, so let's go ahead and try that. Let's see how that looks.
Okay, let me try. I think I cannot share the screen while other participant is sharing.
Oh, so Julia, you have to say stop sharing, and then Nandu can share.
Okay.
Okay, let me share my screen now. Okay, share the screen, and I will say, okay. I need to be selective about what I'm sharing. It doesn't let me just share the screen in general, but the specific program. So you see my screen now, right?
We can. Yeah, it's in, I can see your whole PowerPoint though. Can you click on the presentation though so that it gets big on the screen?
Yeah.
Perfect.
That works well. So in case I need to change it to show something like, for example, let me stop this sharing, and then I will go to go here. Okay, now I can go to the paper, and while here, I also might want to show something like how a USP monograph looks like. So this is the USP online version. So I think I need to stop sharing and then move it. It's not allowing me to share my global screen.
There should be, we don't have a ton of time to diagnose technical issues, but I think what you just showed us was pretty smooth. It looked fine to me, but there usually is an option to share the whole screen, but it sounds like you're familiar and it's not maybe working out well right now.
Yeah. Should be easy.
So, but no, but I think what you showed looked just fine. So.
Yeah, and actually I just saw a setting on here that is allowing me to say all panelists can start sharing when someone else is sharing. So I'm going to turn that on for you.
Okay.
So I don't have to unshare my screen for you to share yours. You can just bring yours up so there isn't that gap.
That sounds great.
Yep.
Sounds good.
Okay. Cool. All right.
Cool. And then I don't know if I wasn't really a part of the prior discussions, but I don't know if there's any complexities to the flow, if there needs to be back and forth, or if we want to leave Nandu's flyer up during questions, or if we want to have Nandu's presentation up during questions. I don't know if you guys talked about any of that.
Yeah, so I think we were planning to just have Nandu's presentation up during the Q&A afterwards. And I'll just give the intro with Nandu's bio, and then I'll ask a couple of questions about Nandu's background a little more in depth for the audience, and then we'll just jump into the presentation from there.
Cool. What I like to do sometimes is when you're asking the background questions to leave the flyer up, so then it's people, because not everyone's listening to every word, so they can read his bio. They're like, oh, that's cool, and then they're reading the bio, and then they're seeing CANN and seeing the flyer. That's just kind of my general practice, but
right. Yeah, I agree. I think that's a good idea. And then Nandu, after a couple of introductory questions, once you respond, then I'll say, okay, with that, I'd like to introduce Nandu's presentation, and that will be your cue to share your screen, bring it up.
So you will start off with the introductory questions that you sent me, Julia?
No, those were the planted questions for after the presentation. So I'll read your bio, and then I'll just ask you a couple questions like, how did you transition within USP to start focusing on cannabis? Something kind of basic, but a little more detail about yourself.
Sure. Sounds good.
Okay.
And then if you guys need me, so I'm gonna turn off my camera And so if you guys need me, I'm here. If for any reason, like Julia, if you're like internet glitches or something, I can jump in. But other than that, I'll just be kind of in the background. So.
Yep. Okay. Sounds good. All right. Awesome. Well,
Nandu, do you have any other questions before we kind of start it?
I think, the standard practice in journal club is to rip the paper apart. Let's do
that's
that.
it. Rip it apart. All right.
Oh man, Nandu, you're getting a little too academic on us. This is for the
That's what I'm
general
expecting.
Really? Do you want me
no.
to call that out to the audience? Say, just so everyone knows,
Oh.
Nandu wants this Q&A session much like in academic journal clubs. So bring the best questions you can.
Seriously, yes. I mean, without
I'll
questions,
say it.
it means nothing really. Yeah.
Yeah.
It should not be a one way. Yeah. So I'm only trying to balance the volume of my presentation. Like, there is so much to talk about it versus allowing time for more questions. So we thought about maybe about 5 to 10 minutes of the intro in the beginning and maybe 40 minutes and then maybe another 10 minutes at the end for questions. But what I can do is to probably condense my presentation so that we can have more time for questions. I can always elaborate on what comes. Really, it would be very nice to have a lot of questions and good discussion.
Yeah, that sounds good. You can whatever time you want to spend on the presentation is fine, and then we'll spend the rest on questions. So Julia, just hit 11, so I'm going to mute my camera, and then if you want to go ahead and open it. Do you have that start webinar thing, Julia?
Yeah, I'm just trying. For some reason, the presentation isn't coming up.
It looked okay how you had it just a moment ago.
Really? Right now?
Not now, maybe 30 seconds ago. But just so people don't think it's not happening, we should start the webinar pretty shortly.
Yeah. All right. Can you see it now? It looks normal?
It does not look normal. It's kind of like zoomed in almost.
Huh. Okay. Let me do this. Share. What about now? No?
No, yeah, it's kind of weird. It's not going into the... It looked fine before. Julia, I don't know if you can like close PowerPoint and just open it again real quick. Might be the move.
Yeah. Okay, let me try that. I don't know why it's not looking normal all of a sudden. Okay, let's see.
It's always these last minute things.
Always, right? Yeah. Okay, go back to Zoom.
Julia, are you opposed to starting this? I just don't want people to think they like can't get in.
Yeah, I just want the presentation.
Oh, there. I think that's going to work now if you click the full screen. We'll click the thing to make it go into like the full screen.
It is on my, it is clicked for that.
That's so weird.
Yeah, let's present.
Julia, do you want me to try to, I know it's like last minute, I can try to share it on my screen. I don't know if that's like confusing.
Yeah, that might be the move because I don't know why suddenly it's not working.
Okay, well, tell you what, let's do this then.
Yeah.
Let's see. Okay, hold on one second. Okay, how does that look? It looks good, right?
Yeah, that's good.
Okay.
So you'll just have to follow my cues.
Yep. Julia, I'm going to go ahead and, or Julia, you click it because you're like the lead. Click start the webinar, but I'll follow your cues.
Okay. So I say broadcast. Yeah. All right.
Go for it.
All right, welcome everyone, and thank you for joining us today for December's Journal Club. So today we have Dr. Nandu Sarma here to talk to us about his Cannabis Inflorescence for Medical Purposes publication with USP, Considerations for Quality Attributes. And so before we get started with the webinar and presentation, I just want to give a few updates and information regarding CANN. So this journal club is brought to you today by the Cannabis Chemistry Subdivision of the American Chemical Society. And so we just have a few announcements to provide to you. So we wanted to let you know to stay tuned on CANN social media for updates on the 2021 Journal Club schedule. So we have a number of speakers we're excited to get scheduled and present to you all in the coming year. So just keep your eyes out for that. And then in addition, we wanted to make everyone aware that the abstract submissions for CANN sessions at the upcoming ACS Spring National Meeting 2021 are open from December 16th to January 19th. And so for that, there are 4 sessions available to which you can submit abstracts. So those sessions are Women in Cannabis, Shaping an Emerging Industry, Advancements in Cannabis Analytical Methods, Cannabis-derived treatments for specific medical conditions, macromolecular interactions and justifications, and lastly, cannabis apart from cannabinoids, molecules from the cannabis plant that will shape the future of humanity. So please do submit to those if you have any interesting research or information you would like to share with the cannabis community. And then lastly, of course, if you aren't already a member of CANN, please do join CANN. We really are entirely supported by our membership, and that's what drives all of the various initiatives that we have going on within our organization. All right. So with that, I would like to also thank our sponsors, because CANN would also not be able to bring much of what we do, including Journal Club, to you without all of our sponsors. Notably, we are within the Division of Chemical Health and Safety of the American Chemical Society, and they, of course, provide our overarching support and administrative support to allow CANN to run. And then for our platinum sponsors, we have Heidolf North America and Carbon Chemistry. For gold sponsorship, we have CFS, and that is run and operated by Jerry King, who is a longtime CANN member and also an emeritus ACS member. So Jerry has a long-standing reputation as a professional chemist. And then lastly, our silver sponsor, Perkin Elmer. So thank you all for your continued sponsorship and support of CANN. All right, so with that, we'll move into introducing Dr. Sarma. So Dr. Nandu Sarma is the Director for Dietary Supplements and Herbal Medicines Program at the USP, responsible for strategy and external stakeholder engagement for new and innovative projects, working with global stakeholders and expert volunteers in the development of quality standards, including monographs and general chapters, for dietary supplements and herbal medicines that are published in the USP Dietary Supplements Compendium and the Herbal Medicine Compendium. Before joining USP in 2006, he was a postdoctoral fellow at the National Cancer Institute in Bethesda and Thomas Jefferson University in Philadelphia, and was a senior scientific officer at the Himalaya Drug Company in India. His research experience includes isolation and analysis of active components of botanicals and their biologic activity. He published more than 25 scientific articles in peer-reviewed journals, and Dr. Sarma holds a pharmacist degree and a Ph.D. in pharmaceutical sciences, pharmacognosy, from Banaras Hindu University in India. So with that, I really extend a warm welcome to Dr. Sarma here. Dr. Sarma and I became acquainted actually through the AOAC Cannabis Analytical Sciences program, and from that have just maintained communication, and I've had the honor of volunteering with Nandu in a number of different organizations, and having Nandu also come and present at one of the CANN sessions this past national meeting at the ACS. So I'm really excited to have the opportunity for Dr. Sarma to present his work and also interact with our members in our greater community here. So before we jump into your presentation, Dr. Sarma, I just wanted to give the audience maybe a little bit more information about your background and how you've transitioned to focus so much on cannabis now. So given that you started with USP in 2006, but cannabis has become a focal point only more recently, can you describe maybe what your experience was initially at USP and how you transitioned into focusing on cannabis more recently?
Sure, I'll be glad to. And thank you for the nice introduction, Julia. It's my pleasure to be here. Thanks for the opportunity to participate in this journal club. I'm looking forward to a very good discussion on the paper. So specifically about me, how I transitioned into cannabis, which obviously has evolved over the recent years. I'm a pharmacognosist by training. All my career, including my master's, Ph.D. was on Indian medicinal plants. So I have a natural interest, experience, and background in botanicals all my life, I would say. And I joined USP in 2006. Obviously, cannabis was nowhere in the picture at the time. We were working on developing monographs, and one of the critical issues was about the admission criteria, what botanicals or what ingredients we can include into the compendia, given the unique regulatory environment related to the dietary supplements. So several botanicals used as dietary supplements. So we were working on the safety evaluation, the contaminant profile, and the appropriate labeling requirements that might be helpful. So we published on the green tea and black kahash about the potential for the liver damage. So that was a lot of interesting work, working with a lot of different stakeholders in that area. In 2016, USP published a stimuli article on the cannabis, following lots of inquiries around that time, and given the increasing usage of the cannabis for medical purposes, and given my background and interest, obviously, I got into cannabis. It's one of my major portfolios at this time.
Great, thank you, Nandu. That was a wonderful explanation. So with that, I'd love to jump into your presentation. So thank you again for being here with us today. I want to give a reminder to the audience that we will have a live Q&A session following the presentation, and you'll be able to directly ask Dr. Sarma any questions you have regarding the presentation and the work that he is presenting. And he himself has said he would love for this to be similar to an academic journal club where you bring him the most difficult questions, tearing apart his paper. So the challenge is all yours. And with that, thank you, Dr. Sarma, and we'll begin the presentation.
Thank you, Julia. Our focus for today's presentation is our recent publication in the Journal of Natural Products. Here you have the paper, and here is the citation. That's a link at the bottom below. I don't know if the PowerPoint slides are provided to the participants. I would be very fine with sharing of this information. So you have a link for that paper, and I will also provide additional links. So I will describe what was discussed in the paper, as well as what we are doing right now after the publication of the paper around March of this year, 2020. I will describe who we are, so that provides a context for this work. The public health rationale that prompted USP's contribution in this area. I will go into the specifics of the components related to the quality parameters for the identity, composition, limit of contaminants, and describe our current ongoing work. For those who are not familiar with USP, USP is a science-based nonprofit organization with a mission to improve global health through public standards and related programs. USP sets quality standards for prescription drugs, dietary supplements, and foods through the input from expert volunteers. USP was founded in 1820. So this year we are celebrating the 200-year anniversary of USP. On a related note, we published a work on the contributions of the USP over the last 200 years to the botanical quality standards. That was published in Herbal Gram in around May of 2020. We compile a lot of information for that, just on a side note. So it started out with 11 founding members who were concerned at the time about the quality of the primarily botanicals at the time, quality of the medicines at the time, and wanted to have a uniform standard for the country. So fast forward to 2020. Now we are over 900 staff and more than 1,000 expert volunteers who are supporting this work. In 1820, there were 207 medicines covered. Now we have more than 6,000 monographs covering not only pharmaceuticals, but also food ingredients, dietary supplements too. There was no formal recognition when it was formed at the time. There was no FDA. And now USP is USP and NF are the official compendia according to the Federal Food Drugs Cosmetics Act with a formal recognition. And the standards are legally also recognized in 38 other countries and used in more than 140 countries around the world. USP was just Pharmacopeia, a book at the time, whereas now it also offers more than 3,600 reference standards, training of the professionals in related topics, and also offers verification and global health impact programs. It was US focused at the time, and now with a very strong global focus, global presence, I would say. So what sets USP apart is that as a science-based organization, USP has a wonderful structure and a legacy of volunteer contribution and participation. So the standards are set by volunteer experts, not by the USP staff. They are all set by the volunteer experts representing international regulators, industry, and healthcare practitioners under strict conflict of interest provisions. There is a lot of collaboration of the external stakeholders, including industry. We organize industry stakeholder forum and organize workshops and roundtable discussions on topics that need further evolution in terms of the science and consensus. We are proud in taking the technical leadership in analytical science, the compendial science related to setting quality standards, all the way from medicine to the food spectrum. USP is governed by a convention representing more than 460 organizational members. So it's a convention of the conventions of the organizations, I would say, which elect the board of trustees, again, volunteers, and the volunteer expert committees that set the standards. So the expert committees are assisted by expert panels. So in this case, the cannabis expert panel was formed to provide the input to the expert committee for setting the standards. In 2020, the USP convention passed a resolution specifically focused on cannabis, which provided mandate to develop fit-for-purpose analytical resources and solutions to help address quality-related concerns and to support additional scientific research on cannabis, cannabis-derived products, and cannabis-related compounds. USP publishes its standards in several compendia, depending on the subject. USP NF is probably the most well-known. USP NF is the official compendium. So that includes several botanical drugs, dietary supplements, biologics, general chapters, and so on. USP also publishes the Food Chemicals Codex. Some of the ingredients could qualify as food ingredients, such as the hemp seed oil. USP Dictionary of the Names, and the Dietary Supplements Compendium, the DSC, and the HMC, which include several botanical monographs. So several of the botanical ingredients used as dietary supplements, such as ginkgo, ginseng, St. John's wort, valerian, and several non-botanicals, they're all available in these compendia. And there is an admission criteria for each of these compendia. USP also provides reference standards, which are thoroughly characterized materials whose validity for usage with the compendial methods has been established through several collaborative studies. Okay, I'll talk about why USP provided the inputs for the cannabis. That is because we are aware through several representations that there is a growing and active interest in cannabis for the purpose of medical usage in the States as well as around the world. And there are also reports of adverse effects from adulteration, contamination, and quality issues. So given the wider usage as well as the growing interest from healthcare practitioners, the researchers, and more importantly, the patients, we recognize that the quality, increased quality standards are quality parameters are important for patient safety. And we recognize that the quality parameters, such as for the identity, test for the composition, and limits on contaminants will help prevent harm to consumers from substandard, adulterated, or misbranded products. So that was the primary impetus. And you may also see that the FDA had recently provided a draft guidance on the quality parameters needed for clinical studies, specifically for cannabis. And we provided comments on that. Besides several reference to several USP journal chapters, such as the chapter 563 for the adulteration, for microbial load, USP rounding rules, and so on. We provided additional information on that. So I'll provide a link to accessing that information later. In 2016, we published a stimuli article highlighting the question of the advisability and feasibility of developing USP standards for cannabis use for medical purposes. So this was primarily to address any public health concerns. That was the primary driver. Following the publication of the stimuli article, we also organized a roundtable discussion, including the industry and the regulatory agencies. And based on the input, we realized that quality standards are required, and that would be helpful for reducing harm to consumers. And USP formed an expert panel to develop the quality parameters. The conversations showed that it is feasible to develop the standards, but it is not advisable to publish those standards in the official compendia. So that was the reason we published as a peer-reviewed article instead of publishing as a USP monograph. So in summary, about the approaches that USP took to the cannabis is consistent with the USP mission. Our initiatives are intended to reduce the harm from products and to enable systematic clinical studies. If we do not take a position about the regulatory status or the potential pathways, we defer them to the regulators. And the most recent publication was in the Journal of Natural Products. So the official USP monograph is not developed, as I noted, due to US regulatory status. And we adhere to the standard compendial processes and admission criteria in determining whether to publish a monograph as a USP monograph, dietary supplement monograph, or maybe or a FCC monograph, or maybe as a publication. So going to the meat of the quality parameters, specifically, a typical USP monograph includes a nomenclature, methods for the identification, and limits for the contaminants, supported by appropriate analytical methods and acceptance criteria. And depending on the intended purpose, we provide the methods that are fit for purpose and that are scientifically valid that meet the USP Journal Chapter 1225 requirements for the validation of the methods for that intended purpose. I take one step back before I get into the identification. This is about the nomenclature. USP Cannabis Expert Panel had reviewed a lot of information on the different varieties of the cannabis. Cannabis is inherently complex, and as I'm sure all the participants are familiar, there are so many different varieties, loosely called strains, but there are different varieties with different chemical profile. And there's a lot of development of well-characterized genomic kind of materials that produce certain kind of a cannabinoid profile compared to other plants that produce a different profile of the cannabinoids. So there was a lot of discussion, and you will find it in the publication, on the nomenclature. USP follows a hierarchical approach with respect to the nomenclature. We provided a guideline, link to the guidelines that we follow. So the title of the article, Cannabis Inflorescence, has a lot of story behind it. I will not tell the story. You can read that in the paper. But what I would like to highlight is that besides giving the name, the paper, you will see, also links the name with the appropriate analytical methods to verify that, and also the acceptance criteria to differentiate it from other names. So I will talk a little bit about the chemotypes that we get to, but the cannabis inflorescence, the title is also associated with additional labeling requirements. So you can see the whole picture and the entire quality as one set of quality. So that is an important take-home message I would like to give here. So in here, what we provided are the macroscopic and microscopic characteristics that can help in the identification of the cannabis inflorescence. So besides the publication, we also published a very detailed supplementary information. So that goes hand in hand, where a lot of information is provided. So we looked into, regarding the identification, we also looked into the chemical profile of cannabinoids, which are of pharmacological significance. You know the CBD, THC, but cannabis also produces a lot of other components, and new varieties are coming up with time that are rich in different cannabinoid profiles. So we looked at the different cannabinoids for reason also that whatever methods are proposed should be able to differentiate and resolve the different cannabinoids so that there is not a underestimation or overestimation of the cannabinoids because of the overlapping peaks and lack of separation. So that's an important consideration. So in here, what is provided is a TLC method for the identification. So identification, again, you need a specificity according to the Journal Chapter 1225. And so you need to identify that it is cannabis and differentiate from other closely related species. And within cannabis, there are 3 different chemotypes that the Cannabis Expert Panel had identified: the THC dominant variety, the THC CBD intermediate variety, and the CBD dominant chemotype. So here you can see that the major cannabinoids are they do separate. I agree, and I totally recognize, yes, you do have certain closely migrating bands that can essentially overlap. But what's important is that they do separate when they are decarboxylated. So for example, This is the THC dominant variety. So in the native form, before decarboxylation, you would see THCA, which when decarboxylated will get into the THC, will becomes THC, and thus it will, you can have a profile searching by both the ways, and you can identify that. Similarly, in the middle, you have the set that is the intermediate variety before and after decarboxylation, and the CBD dominant variety before and after decarboxylation. And there is a lot of discussion in the paper. Another important topic I would like to highlight here is that with each of these methods, they are linked to a specific journal chapter, like 203 and 1064 in this case. So compliance with the journal chapter is really important because TLC is notorious. It may not be reproducible unless you have all the conditions appropriately controlled. So I invite you to read closely the 1064, which gives with illustrations as to what happens if you don't control the, like humidity, or, what happens if you spot it not 8 millimeters from the bottom, but, someone else spots 2 centimeters from the bottom, and so on. Or you don't run 6 centimeters of the resolution, or rather, what happens if you go to 10 and then you try to compare the two different results. So we have a lot of illustrations there. Similarly, HPLC method is provided both for the identification based on the fingerprint peak matching, and also for the quantitation. The chromatographic conditions are provided here. There's a lot of information in the paper about the challenges that we faced and how to resolve them depending on the specific needs. So we used the formic acid based mobile phase and used the C18 column. I can go into the details if there is a further interest. We used the method with buffer. I just wanted to illustrate, but there are challenges, and even with a very fine pH difference, you could have overlapping peaks, whereas with a non-buffer method, we got very good resolution. And we provided additional ways by which some of these overlapping peaks could be resolved. For example, we specifically noted that the delta 8 THC and the CBNA, we recognize that does overlap. And what we noticed through a lot of literature that both delta 8 THC and the CBNA are very minor component. Their presence is really not observed. So there may be possibilities, some of the varieties might contain them. So if you see a peak there, and if you would like to resolve, you can use 5 millimolar ammonium formate, and you can have a nice separation of the delta 8 THC and the CBNA. So there is a lot of discussion there. Similarly, we provided the GC method. You would have to go to the supplementary information for additional information. It's not in the main paper. All these details are in the supplementary information. So this is the GCFID method is provided based on the derivatization with the TMFs, and could get good resolution. We did some work on the stability of the reference standards. The carboxylated cannabinoids are very susceptible to the temperature. They get degraded with time, depending on the temperature. So we recognize that it needs a stabilizer. So we did a very quick, very short time study at 20, 25, and 40 degrees, and you can see a very clear high degree of impurity generation in no time. So it needs a stabilizer. We tried different conditions, including aprotic solvent, acetonitrile, and triethylamine, the BHA, butylated hydroxyanisole, and ascorbic acid, and found that BHA is not as helpful with the TEA and ascorbic acid is much better. We ran additional studies and found that the optimized conditions are when, and we provided the information in the paper, 68% of the acetonitrile, 32% of the triethylamine, with 0.15% of the ascorbic acid. So we provided those conditions as stabilizers. So besides the purified compounds, either as solution like the THC 1 mg per ml in methanol, CBD 1 mg per methanol, and the CBD as a pure compound, we also provided two unique mixtures of the reference standards, the cannabinoid acids mixture with in acetonitrile and with stabilizers, and the cannabinoids mixture in methanol. So depending on the stability, two unique sets of the cannabinoid mixtures are offered as reference standards, and their usage is referenced in the methods which are provided. So I talked about the three chemotypes, the THC dominant, CBD dominant, and the intermediate. So what you can see is that there is a specific acceptance criteria associated with each of these names, which include that for a variety to be called a THC dominant cannabis chemotype, it should exhibit a principal peak for the THCA. The ratio of the total THC, again, as many of you are already familiar, the total THC is the THC plus THCA content. To the total CBD is not less than 5:1 and contains not more than 1% of the CBD and not less than 1% of the THC. So you can see very similar acceptance criteria for the CBD dominant with the inverse ratio of the total THC to CBD and inverse ratio of the CBD and THC. Again, all these criteria are in the paper. Similarly, for the intermediate variety also, there is a clear expectation of the criteria for meeting that particular nomenclature. The paper also deliberated upon what is the allowable variance that is possible. And you can see in USP, if it is ascorbic acid as a monograph, it is not less than 99.5%. So if ascorbic acid is less than 99.5%, it's not a USP grade ascorbic acid. We know not less than 99.5% of the expected cannabinoids is not practical, reasonable for a botanical. Not less than 80% is also not reasonable. So depending on the specific matrix and based on lots of data, the allowable range is not less than 80 and not more than 120% of the labeled amount in milligrams per gram of the total THC and the CBD. Similarly, there is also expectation to label other cannabinoids which are present in an amount of 1% or more, the 10 mg per gram or more. So if there is other cannabinoids, that should also be labeled. And for the THC dominant chemotype, the content of the CBN, which is a marker for the degradation, is not more than 2% of the content of the total THC. The paper also provides methods and acceptance criteria for the dominant and co-dominant terpenes based on analysis of lot of data. We recognize that D-limonene, beta-myrcene, gamma-terpinolene, alpha-pinene, and beta-caryophyllene are the dominant terpenes. And in some cases, there is a codominance of these terpenes. So considering the limited evidence that terpenes may contribute to the known clinical effects and to facilitate appropriate clinical study, we have provided ways to label the terpenes so that their contribution could be appropriately investigated. Okay, limits on contaminants. There's a lot to be said about, I think I need to cut down on my discussion, but I'll be happy to answer any questions. There's a lot to be said about the limits for the pesticide residues, especially given the possibility of incidental contamination through pesticides which are not directly applied, but maybe through the soil and through the drift, environmental drift. A lot of discussion went into providing a risk-based and stringent requirement of the limits for the pesticides. So I can talk more if there is an interest on that. Similarly, cannabis is known to be a hyperaccumulator of the elemental impurities. So the panel recommended for adoption of the limits from the USP General Chapter 232, elemental impurities, and the limits for the inhalation products. So that is for the major elemental contaminants. Similarly, for the microbial contaminants, there is a lot of discussion in the paper. I will not go into the details for paucity of time. There is another level of the limits for the microbial contaminants is provided, which is for cannabis, which is specifically either treated or generated in such a way that there is a reduced microbial load, meeting these requirements, which might be suitable for inhalation purpose, specifically in immunocompromised individuals. So the limits from the USP General Chapter 1111 for inhalation use are recommended with appropriate link to the labeling also. Aspergillus, there were no methods that the panel could find. So there was a guidance provided in terms of the USP General Chapter 1223 on the validation of the alternative microbial methods. The AOAC is working on, they developed a SMPR and working on methods, so that would be helpful for the community. So these guidelines provide for the appropriate processes for testing the microbial recovery and identification, and the validation characteristics such as the accuracy, precision, specificity, detection, quantitation, and so on. There are two areas that the paper had identified in terms of research gaps, and one of them is the aspergillus, and other one is about the minor cannabinoids. So this was one of the things that was highlighted in as a research gap in this space. A limit for mycotoxins, a reference is made to General Chapter 561 for the methods and the limits for the aflatoxins. Not more than 20 ppb for the total aflatoxins, and not more than 5 ppb for the aflatoxin B1. There are additional recommendations for the sampling, which is a very important one in order to make sure that the sample is representative of the lot. Recommendations about the water activity, we adopted the recommendations from the ASTM about the water activity control, so that it is not, so that the material is not too wet to promote degradation, or not too dry to avoid degradation and decarboxylation. And we provided other guidelines to help accomplish that objective. We provided relevant information related to the control of the foreign organic matter, ash value, and the packaging and labeling. Similarly, there is a section on the labeling. I will not go into that. It is all described in the paper. So I'll talk a little bit about what we are doing now, which is not captured in the paper. Right now, we are collecting information in order to develop appropriate quality attributes for the cannabis extract. So we worked on cannabis flower. Now the next is cannabis extract, hemp and hemp extract, and the CBD, consistent with USP admission criteria. Regarding hemp, you know the definition of the hemp. It is a cannabis with all the associated definition requirements and not more than 0.3% of the delta-9 THC. So there are 3 GRAS notices without any questions from the agency, specifically the hemp seed protein, and the hemp seed oil. So the FCC, the Food Chemicals Codex team at USP, is working on the hemp seed protein and hemp seed oil monographs. They are under development at this time. When it comes to hemp, there are certain unique challenges. Again, challenges related to the nomenclature. Think about all the, again, different varieties of hemp similar to the cannabis. So what is the variability within that? We are collecting information on that. There is more information from Canada than from US, and with the new development, things are not as evolved. So we are at the phase of collecting information. And we will follow similar compendial approaches, for example, fingerprint for naturally occurring cannabinoids, so that you can identify that it is hemp. Limit of not more than 0.3% of the delta-9 THC. And what about the limits for the cannabinoids or the acceptance criteria for the cannabinoids? We are collecting information on that, specific tests for the terpenes, and then limits for the contaminants. In case of extracts, we will take the risk-based approach from residual solvents, General Chapter 467. And when it comes to extracts, again, there are a lot of complexities related to the different kinds of extracts that are available. There's a lot of innovation in this area. So we need to be considerate of them and make sure that they are named appropriately, and they could be differentiated appropriately, so that there is a clarity of communication between the dispenser or the healthcare practitioner, the researcher, and the patient. So you have broad spectrum, full spectrum distillates, isolates, and so on. So this is an area that we are working on. So in the end, we believe that public quality standards can help in protecting the patient from harm, and they can help in monitoring the product quality with appropriate titles, useful definitions, validated methods, and established limits. So here is my email. I'd be happy to receive any questions. And here is a landing page for USP Cannabis topic, where you can download the paper, you can get USP comments to the FDA and USDA, relevant USP general chapters, and reference standards. Thank you all.
Thank you so much, Nandu. That was a wonderful presentation, and we've had quite a number of questions come in, so I'll get to those in just a moment here. So to start off, what is the difference between a USP general chapter and monograph?
So it is about the compendial hierarchy. Monograph is specific for an ingredient. For example, cannabis has a monograph that defines all attributes related to that particular matrix, such as What is the appropriate test for that particular matrix? What is the test for that ingredient? And what is the limit as appropriate based on information for the contaminants, labeling, and all those things. General chapter provides the information which is common for several monographs. For example, General Chapter 203 is for the HPTLC, 621 is for the chromatography. So we might refer the chromatography chapter as appropriate. So when someone is conducting the test, they can use all that information, including, for example, in case of HPLC, if you want to modify from HPLC to UPLC, so you can use that depending on certain variables, and it provides a guidance how you can change while still staying within the system suitability requirements and so on. So general chapter is a broader a description of a specific activity that may be applicable to several ingredients. And they are called in the monographs.
Great, thank you for that thorough response. I know that even in my work from day to day, it's really helpful for me to understand the breakdowns of those various documents, so I appreciate that context. So we have a question from Cody Peterson that kind of falls in line with what we were just discussing. Do products made from cannabis inflorescence, for example, extracts, fall under USP non-sterile compounding guidelines? What USP standards exist for cannabis manufacturers, growers, etc.? If none, is there a plan to develop?
So we did not discuss the applicability of the compounding guidelines to to this cannabis space. So I would not be able to answer a question about the compounding chapter's applicability. It's a huge area, a huge group, and it has its own applicability. For example, the compendium chapters, the compounding chapters are not applicable to the dietary supplements area. If someone wants to compound a ginkgo extract for a certain specific purpose. So I would not be able to answer that question. But could you repeat the question? I'm not sure if I missed it.
Yeah, of course. So in the second part of the question, it was then asked what USP standards exist for cannabis manufacturers or growers, and if none, is there a plan to develop them?
So the publication is our avenue for putting the recommendations for the cannabis. We did not produce a USP monograph because of the unique federal regulations associated with cannabis. And it's not a dietary supplement either. So given the unique regulatory environment, the publication is the avenue that we took by which we provided the recommendation. So it's not a USP official monograph, but this is an additional avenue by which we provided the USP expert committee recommendations.
Okay, great. Thank you for that response. So now I'm just going to jump into some of the more technical questions that we saw come through as you were presenting. And I believe this question was with regard to the stability study slides that you had displayed. And so an anonymous attendee asked, have you noticed CBD metabolize into THC in any of those studies?
CBD metabolized into THC? No, we did not see that. We did not notice that. Yeah, I think I am aware it is technically possible. Mm-hmm. I think I recommend reading Jeremy Melanson's publication. I don't know if it has already come out, but he did a very good work on the stability of the cannabis flower, and including the possibility to date the shelf life based on the cannabinoid profile. So it's a very good work.
Right. Okay. Great. And in that same topic, do you think TEA can be used to enhance the stability of products for human consumption?
I'm sorry, you said...
can TEA be used to enhance the stability of products for human consumption?
I don't have any comments about that. I'm not sure if TA could be used at levels which are needed for human consumption. So I cannot say that it can be. I'm sure the USP General Chapter 467 has limits to all the solvents, including the TEA. So I refer to that as probably the limit up to which they can go.
Right. Okay, that's very helpful. Thank you. So then Ramesh Jagannathan also asked, with regard to the chemotypes that were being displayed, on what rationale is a 5-to-1 ratio fixed for THC to CBD or CBD to THC mixtures?
That's in fact a very good question. We have analyzed lots of data, and what we found was that 5-to-1 is the minimum ratio that is present to define a chemotype. But what we noticed and we said in the paper was that typically you will see something like 20-to-1 or maybe even 200-to-1 of the ratio. So predominantly, if you look at the genetics, the genetic composition, if a particular variety is designed to produce the THC, then it may not produce the CBD at all. So in the sense it is so predominantly skewed to produce one cannabinoid compared to the other through the genomic pathway. So typically you will see that it is very predominantly skewed to one or the other, but 5-to-1 was at least the minimum that we could see. So it was based on analysis of a lot of data.
Well, that's very interesting, but I think is a very good point. And we tend to see even in the regulated marijuana industry that to your point, we'll see most cultivars are very predominantly either THC or very predominantly CBD. There isn't as much of that equal split ratio. Yeah.
Yes.
So in General Chapter 561, is that an appropriate chapter to reference for hemp chemotypes at this point?
General Chapter 561 is articles of botanical origin. No, it is not specific to define the hemp chemotypes. General Chapter 563 is the chapter on the identification of the botanicals, of articles of botanical origin. That includes certain generic methods. Again, that includes the microscopy, macroscopy, the chromatography method, and the DNA barcode method is also included in that. However, that they are all generic methods. They are not specific. For hemp, to define, again, we don't know what is the chemotype. So if you look at the broader chemotypes, hemp will fall within the CBD dominant chemotype. But the way it is defined is that it contains less than 1% of the THC. So it is a subset of the CBD dominant chemotype. And there are a lot of variability. So we are collecting information to see if there are subtypes or whether hemp as one set can be defined. So 561 is not the right one to define it specifically. We need data specifically for hemp to set that acceptance criteria.
Thank you for that very thorough response. Yes, that makes sense. And I'm sure as the various industries develop, there may be, chapters more specific to cannabis one day, but it makes sense that as it stands now, it's not as specified for hemp. Yeah, so a more analytical method-based question here from Ramesh again. So what is your opinion on using LCMS for analysis of cannabinoids and subsequent data analysis using pattern recognition methods?
Oh, definitely, yes. Yeah, very, very good thought. And in the paper, we made a reference to maybe opting for LCMS where there are any questions, for example, the coelution of the delta 8 THC and the CBNA. You could use UV as a way to resolve them. And you could use 5 millimolar ammonium formate to separate and then use UV for resolving, or maybe the LCMS as a way to identify them. So we need more methods. So there is more of evolution needed. More refined methods are better, and with time we would love to adopt more methods and better methods in what we publish too. Yeah, it's a very good idea. Yeah.
Yeah, I absolutely agree. And I think, again, as the industry develops and matures, we're seeing more and more methods applicable to the same analysis types. And there's a lot of exploration, and I think more fine-tuned applicability we can locate there. So it's exciting, but I agree that is a great suggestion. So now we're switching over to some questions around contaminants to expand on that a bit. So Robert Thomas asked, why only specify 4 elemental contaminants when it's universally recognized that many more elements could potentially be absorbed by the plant during the cultivation process? And I know this is a topic that's been coming up recently when considering regulations in Colorado as well as some other states, and with everyone really trying to figure out which elemental contaminants should be included in those testing panels.
Yeah, so what the panel did was to take a risk-based approach, and obviously these 4 are the ones which are the big 4 where there is more of a concern. But we also recognized in the paper that if there is a possibility that additional contaminants are possible, let me just quickly see the section. So this is the section on the elemental contaminants. We said that when additional elemental impurities are known to be present, have been added, or have the potential for introduction through whichever means, assurance with the specified levels is required. So this is part of the general chapter 232. So listing of these 4 elements based on risk does not imply that any other elements at much elevated levels are acceptable. That does not imply that. So where there is information based on the risk, based on the soil type, water type, additional testing may have to be done.
Absolutely, I agree with that. And that's something regulatory bodies encounter quite frequently as well. And I know we're at the top of the hour, so we are coming to the end of our presentation here. So I'll just ask you one last question that ties into that discussion surrounding contaminants and how that combines with considerations for regulators. And so this comes from Yvonne Depore. How would regulators be able to detect pesticides that are not on their own list?
Very good question. In fact, we discussed this quite a bit in the paper too. I strongly encourage to take a look at that. There are several regulations which specified either the pesticides to be limited or kind of pesticide that should not be present, but a whole lot of other pesticides could be present because of the environmental drift or the soil conditions and so on, even though they are not applied. So what you see in those cases is that they are at much reduced levels, but above the levels which might prompt for control. For example, 10 ppb is kind of a de facto tolerance level for the pesticides. There may be pesticides which are above that level, but they may not be really of safety concern. For example, some of these pesticides are allowed at levels much higher than that in other foods, like blueberries and so on, but it's zero tolerance for the others. So there is a lot of discussion. We organized a roundtable topic on this pesticide-specific topic. We wrote a stimuli article. I'd be happy to provide a lot more information if someone sends an email on this topic. But so what we provided in the paper was that besides limiting whatever is the requirement for the state by the state, the other pesticides also should be within limits of based on the toxicological considerations. So we provided a formula for that, and we discussed that in the paper. So we did consider this particular possibility.
Great, thank you so much. Well, with that, we are at the conclusion of our presentation. So thank you, Dr. Sarma, for the wonderful presentation and very in-depth Q&A session. That's greatly appreciated. And thank you to our audience for attending today and providing so many insightful questions. This has been a really great discussion. And with that, we appreciate your support of CANN, and we look forward to seeing you next time. Thanks so much.
Thank you all. Bye-bye.